So why am I going to be talking about T follicular helper cells today? Well, I think you saw this slide from Barney, and I think the real question that we're trying to answer is what will it take to get an HIV vaccine that works like these vaccines, which obviously have great efficacy. As you heard from Barney, we're probably going to need antibodies. We're probably going to need broadly neutralizing antibodies. And as he pointed out, there are several targets of broadly neutralizing antibodies, but they all have certain characteristics, which is going to make them difficult to stimulate through a vaccination. First, they have these long CDRH3 loops, except for the CD4 binding site antibodies, and there's not a lot we are going to be able to do about that. That's going to be encoded within the naive B-cell precursors, and we're just going to have to overcome the low precursor frequency of those long CDRH3s. But they also have extensive somatic hypermutation, meaning that they've gone through a lot of affinity maturation. And in order to overcome that obstacle, we're going to need to engage follicular T helper cells, or T follicular helper cells. Well, what are T follicular helper cells? Well, they are these T cells which are in the lymph nodes. They are not in the peripheral blood. And they actually migrate into the B-cell follicles and into the germinal centers where they interact with B cells. They do this by upregulating CXCR5 and downregulating CCR7, which allows them to move out of the T cell zones and into the B cell zones of the lymph node and into the germinal centers, where through a number of different interactions that are all regulated through the master regulator BCL6, they interact with germinal center B cells, they liberate IL-2, they interact through ICOS, the SLAM family members, high PD-1, and other interactions. And through these interactions, they induce within the germinal center B cells activation-induced deaminase. And this is the key enzyme that's involved in inducing somatic hypermutation. So in order to actually get extensive somatic hypermutation, we're going to have to engage these cells. What I will talk about today are several things. Because these cells are present in lymph node, they can be difficult to evaluate in humans, so we've spent a lot of time looking at these cells within rhesus macaques, where it's easier to access the lymph nodes and easier to time our experimentation. So I'll show you the characterization of these cells in rhesus monkeys, the impact of SIV infection, how they're associated with broad neutralizing antibodies rather than a SIV, a SIV model, and then how we can induce these responses in vaccination. And then I'll tell you some work that's been done on whether there is an equivalent of these lymph node-associated T follicular helper cells in the periphery, and that will be work that has been done in humans. So first, in order to assess T follicular helper cells in rhesus monkeys, we came up with a flow cytometry panel where we gate on CD3 cells. These are all CD4 cells, so we gate on CD4 positive, CD8 negative cells. And then we really want to look at the central memory type of cells, which in rhesus macaques are well defined with two markers, CD28 and CD95, so this population. When we gate on those, what we're looking for are the cells that have downregulated CCR7 and upregulated PD-1, and so that's this population right here that are CCR7 low and PD-1 high. To further confirm that those are probably the T follicular helper cells, we can look at a couple of other markers, ICOS, which should be upregulated, and as you see on these cells here, they have high ICOS. These other cells are lower in ICOS. In addition, as they move into the germinal center, they should start to downregulate SLAM or CD150, and what you see is that these cells tend to be lower in CD150 than are these cells. We can then stimulate these different populations that are color-coded here and ask what cytokines do they produce. And you see, if we look at the cells that are not T follicular helper cells, they make interferon gamma, TNF, IL-2, a little bit of IL-4 and IL-17. So that these cells here are really your Th1, your Th2, and your Th17 cells. If, however, we look at the cells in blue and black that we're assuming are T follicular helper cells, they make lower interferon, TNF, and IL-2. They actually, the cells in black, the CD150 low cells, actually make increased IL-4, which is one of the characteristics of a T follicular helper cell. And they don't make any of the Th17 cytokine IL-17. The ability through Luminex to measure monkey IL-21, which is pathognomonic for T follicular helper cells, was not good, so we had to go to RT-PCR. And what you can see is that the cells that make the highest levels of IL-21 are indeed these cells here. So throughout the rest of the talk, I'll refer to any CCR7 low, PD1 high cell as a T follicular helper cell. If they're CD150 or SLAM high, they're just a general T follicular helper cell. If they're SLAM low, I'll call them germinal center T follicular helper cells. Those are the ones that make higher IL-21, higher IL-4. Just functionally, we can then co-cultivate these different cell populations with B cells and ask, do they actually support B cell differentiation? And this simply shows you that this population actually does much better at supporting B cell differentiation, which is what a T follicular helper cell should do. They should also express the master regulator BCL6, and this shows that indeed they do. But really, to define a T follicular helper cell, it should be in the germinal center. And so we used confocal microscopy using 4 different colors to identify these structures that look like germinal center. What you see is that within these germinal centers, there are a lot of cells that are very high for PD1. Those cells, it's difficult to see here, but those cells do co-express CD4, but they don't express Ki67. And the cells that are expressing Ki67 are actually B cells. So this is exactly what has been described in humans and in mice, that within these germinal centers you have a light zone where T follicular helper cells interact with B cells up here, and then you have a dark zone where the B cells then move and proliferate, and you see that here as B cells expressing Ki67. And PD1 high is a very good indication of cells, CD4 T cells within the germinal center, because here what you can see is that you can actually quantify how close these cells are to the center of a germinal center, and the cells that express high PD1 are actually closer than those that are low for PD1. So what happens with SIV infection? Now, usually with SIV infection, you get infection and depletion of CD4 T cells, and here I show you monkeys that are not infected, that are acutely infected, or that are chronically infected. And these are your T follicular helper cells. And interestingly, instead of these cells going down, there's actually relative accumulation in some of the monkeys. And what you can see is about half the monkeys are very similar in terms of their percentage of T follicular helper cells to uninfected monkeys, but about half of them actually have relative accumulation of T follicular helper cells. So we can just show that again here, where now in red you have the monkeys that have high percentage of T follicular helper cells, and in black those that have a normal percentage of T follicular helper cells. And what you see is most of that accumulation is within this CD150 low or germinal center T follicular helper cell population. So T follicular helper cells are not being depleted, they're actually accumulating during SIV infection. And what that becomes in the lymph node are these very large hyperplastic lymph nodes with great big germinal centers, just jam-packed with CD4 positive, PD1 positive T follicular helper cells and proliferating B cells. And those of us who are old enough to have seen patients early in the epidemic will remember that the way we made the diagnosis was lymphadenopathy quite often. And that's what you're seeing here. That lymphadenopathy is this germinal center hyperplasia. As you heard from Hendrik Streeck yesterday, this is not a phenomenon that is isolated and specific to SIV infection in monkeys. The same thing happens with HIV infection in humans. So what is actually driving that accumulation? In the interest of time, I'm not going to take you through all the data. It is published, but I'll just take you through a cartoon of what we have found is happening. So one possibility as to why these cells are accumulating is because they're not getting infected with SIV, and so they can remain, whereas all the other cells, CD4 T cells, are getting infected. And that's not the case. All of the CD4 T cells in the lymph node appear to be equally infected with SIV. How about turnover? Well, there's fast turnover of these cells. It tends to slow down a bit when you get into the T follicular helper cells, but those cells are also very highly susceptible to spontaneous cell death. So we don't think they're accumulating because they're resting in the germinal center and not dying. So the only thing that was really left is that SIV was somehow driving the differentiation of T follicular helper cells. And through a series of experiments, we showed that that's exactly what's happening, probably somewhat in an antigen-specific manner, but also from the general immune activation that occurs in SIV and HIV infection. You get increase in IL-6, which is known to be a factor that drives the differentiation of T follicular helper cells. We showed that there was actually altered STAT phosphorylation within the T follicular helper cells so that you were getting more phosphorylation of STAT3 instead of STAT1, and that drives the differentiation of T cells towards T follicular helper cells and away from Th1 cells. So these data were published late last year in Journal of Clinical Investigation, and I would ask you to go there for more information. In addition, when you get a lot of T follicular helper cells, you should also get a lot of B cells, and that's exactly what happens. So we can evaluate in these SIV-infected monkeys their B cells in the lymph node. You can look at germinal center B cells by looking at their ability to bind peanut agglutinin, and what you see is that there are B cells that do this within the lymph node, not in the peripheral blood. When we look at SIV-infected monkeys with high T follicular helper cells, they have a higher percentage of these B cells. They express higher levels of BCL6, and these monkeys have higher levels of immunoglobulin. So rhesus monkeys' T follicular helper cells have been defined based upon surface phenotype, location within germinal centers, and cytokine and RNA transcript expression. SIV leads to an accumulation of these cells, and it's predominantly within the germinal center T follicular helper cell subset. And accumulation of these cells is associated with more germinal center B cells with high BCL6 and higher titers of SIV-specific antibodies. Now, obviously, if all of this immune activation and all this time is required, it's going to be very difficult to induce this through vaccination. So what we really want is a model where we can now start to assess what it takes to drive the expansion of T follicular helper cells and the induction of broad neutralizing antibodies. And I think there is probably a good model. Recently, Dennis Burton and Mal Martin have shown that a SIV, SIV88, which is based upon the envelope of HIV ADA, when you infect monkeys with this SIV, you actually can get the induction of broadly neutralizing HIV-specific monoclonal antibodies. So we feel that SIV88 infection in macaques may be used to study the relationship between T follicular helper cells and broad neutralizing antibodies. So we took 9 monkeys, we infected them with SIV88, and then we did lymph node biopsies before and then at multiple time points after initial infection. And what you see is all of the monkeys became infected. They all started and had variable loss of CD4 T cells. And if you look then within the lymph node at CD4 T cell loss, you see that they all tended to lose CD4 T cells, except at about week 20, the central memory CD4 T cell population started to expand. And as you can probably guess, that's because starting at week 20, we started to see this accumulation of T follicular helper cells within that central memory CD4 T cell population within the lymph node. So this looks as though it's modeling what happens in HIV infection and SIV infection. So was this accumulation of T follicular helper cells associated with an accumulation of B cells? So once again, we can look at either the total within the lymph node, the total IgG positive B cell population, or those that bind peanut agglutinin or the germinal center B cells. And sure enough, what you can see is if you compare accumulation of T follicular helper cells in these monkeys with the total IgG memory B cell population in the lymph node, there's a very nice correlation. And you can do the same thing for the germinal center T follicular helper cells and the germinal center IgG positive B cells. So, and this is a common theme, that if you get T follicular helper cells, you're going to have B cells. They go together. You see them when you have lymphadenopathy and germinal center hyperplasia. But what about antigen-specific responses? How can we measure those? Well, we can detect on specific memory B cells in the lymph node, and the way we do that is to gate on your IgG positive memory cells in the lymph node. And then we use a probe, an envelope probe, that's fluorescently labeled, so any B cell that has an immunoglobulin that will bind the envelope will stain positively. And what you can see is pre-infection, you don't get many or any of those cells, but by week 44, you start to find these antigen-specific, envelope-specific B cells in the lymph node. By the same token, we can look at antigen-specific T follicular helper cells by stimulating the cells with either envelope or GAG, and then gate on the phenotype of a T follicular helper cell in the lymph node. And what would be best is to measure IL-21 production. Unfortunately, the antibody for monkeys doesn't work very well, so instead we use expression of CD154 or IL-4 as a marker of responding cells. And finally, in these monkeys, we can look at broad neutralizing antibodies. So here's a panel of 15 different viruses. Red is these are the monkeys over here, and red is very good neutralization against that isolate. Yellow is intermediate, green is low, and you can total that up, giving a red a score of 3, yellow 2, and green 1, and you can come up with a sort of an amount of breadth of neutralizing antibody. And then you can compare that neutralization breadth here to the unspecific germinal center B cells, and there's an excellent correlation. So when you have envelope-specific germinal center B cells in these monkeys, you get great neutralization. If you just go to the memory B cells in the lymph node, that's not as good. The important thing here is that if you look in the periphery, you're wasting your time. There's no correlation between the memory B cells in the periphery and broad neutralization. It's all happening in the lymph node. And for T follicular helper cells, looking at specific T follicular helper cells in the germinal center, you get a reasonably good correlation with neutralization breadth, whether you look by CD154 or IL-4. If you look at a Th1 type of cytokine, interferon gamma, you don't see this correlation. So SIV88 infection can be used to study factors associated with the generation of broadly neutralizing antibodies, and the frequency of on-specific germinal center B cells correlates with broad neutralization. And that really, the correlation is with germinal center B cells, but don't look in the periphery and expect to get anywhere. Once again, T follicular helper cells in the lymph node correlate with neutralization breadth, and that's true if you look at CD154 or IL-4 responses. Probably in humans, if you looked at IL-21, you'd see the same thing. And I showed you the on-specific response. You can see the same thing with GAG. Hendrik presented yesterday that GAG responses were better than ONV. We can look at either within the monkey, and we see these good correlations. But once again, you have to use a cytokine that's associated with T follicular helper cells. If you use interferon gamma, you'll actually see, if anything, an inverse correlation. So ultimately, what we want to know is how are we going to be able to stimulate these types of responses through vaccination? So in order to address this issue, we collaborated with one of my colleagues at the Vaccine Research Center, Bob Cedar, who has been looking at adjuvants. And we tried to address the question, are certain adjuvants better than others at inducing T follicular helper cell responses? In this study, he had groups of 6 monkeys which received 4 separate vaccinations with a clade C HIV envelope trimer. These monkeys were either given envelope alone or envelope with multiple different adjuvants, alum with TLR4, alum with TLR7, MF59, et cetera, et cetera. I won't show you all of the data because we didn't do lymph node biopsies on all of them. We simply chose some of the monkeys that had received some of the adjuvants, and those are envelope plus alum and TLR7, envelope plus MF59, and poly-IC-LC. These are the results of the antibodies from these monkeys. So these are the binding antibodies, and these are the neutralization titers against tier 1. And these are just the monkeys that we studied. And what you can see is envelope alone doesn't give very good binding or neutralization. Alum does better, and this dotted line is the average response that you get for alum. And then you see these other adjuvants, alum TLR7, MF59, or poly-IC-LC, actually give you better binding and neutralization, but all about the same. So then what we did is we went back to these monkeys, and 10 days after their last boost, we did lymph node biopsy to look for T follicular helper cells. And why did we choose 10 days? Because we actually did a MF59 vaccination and then serial biopsies to find out when was the peak of the T follicular helper cell response in a monkey. And what you can see is pre-vaccination, you don't have these cells, and then they start to show up, and then they start to go down. And the peak of that response is somewhere between 7 and 10 days, whether you're talking about total T follicular helper cells or your germinal center T follicular helper cells. So we looked at 10 days after vaccination for the accumulation of T follicular helper cells. The way this study was done was quite nice in that each monkey was vaccinated on one side with PBS and the other with the vaccine plus the adjuvant. So you could take lymph nodes from either side and compare them as an internal control. And what you see is that here's the sort of the baseline level of T follicular helper cells in the lymph nodes from these monkeys, and here's on the vaccine side what you get. And what you see is that alum TLR7 gave the biggest increase in T follicular helper cells. MF59 did fairly well. poly-IC did not do well either for total T follicular helper cells or germinal center T follicular helper cells. And that's actually not unexpected, since poly-IC-LC really stimulates a Th1 response. So remember, you've got differences here now in terms of how well these different vaccines induce T follicular helper cells. But all three of these adjuvants gave very similar binding antibody and neutralizing antibody. So obviously you got good binding antibody here with the poly-IC-LC in the absence of stimulating T follicular helper cells. And then finally, we could also look in these lymph nodes at germinal center B cells. And what you see is that the germinal center B cells also vary by the adjuvant, as do the T follicular helper cells. And across all of these different adjuvants, there was a reasonable correlation between the frequency of germinal center B cells and T follicular helper cells. So protein adjuvant vaccinations do lead to a transient increase in T follicular helper cells in draining lymph node, and expansion is dependent upon the adjuvant used, but it doesn't really correlate with overall binding or neutralizing antibody titers. And this may have more to do with the types of envelope antigens that were used. And as you heard from Barney, we may need to vary the envelope over time in order to induce further differentiation of the B cell response. Not all adjuvant effects that result in a better antibody response are T follicular helper cell dependent, and we're evaluating antigen-specific germinal center B cells and T follicular helper cells. I only showed you total, but we have better probes now. So now what I'd like to do is switch gears to what's been called the peripheral T follicular helper cells in humans. A lot of people feel that it will be important to monitor T follicular helper cells in vaccines, but they're very worried about having to do lymph node biopsies in order to do that. And so they've been searching for a way of looking in the peripheral blood and saying, here's what's going on in the lymph node. Well, a couple of recent publications, one of which is here, have identified a CXCR5 positive CD4 positive T cell in the periphery that looks like the peripheral equivalent of T follicular helper cells. What this paper says is that these cells do exist within the peripheral blood, but that this set of markers does not actually fully define peripheral T follicular helper cells. I mean, these cells, this population also contains Th1 cells, Th2 cells, and Th17 cells. So it's not a really pure population. And these can be further characterized using other markers such as CCR6 and CXCR3. So we went on to try and further characterize and also confirm these results. So what we did is took peripheral blood from individuals, and then we looked at the naive cells in red, the central memory in green, or the effector memory in blue, further characterizing those with CCR7. As expected, the central memory cells were mostly CCR7 positive, not the effector population, but the naive cells were. If you then look at which of these populations actually express CXCR5, the important marker for TFH, it's really all of these central memory cells that are CCR7 positive, where the CXCR5 positive cells are contained. And then we went on to further characterize these by CCR6 and PD1, breaking them into these different populations. Now, the first thing that we wanted to do is these functional assays of if you take these different sorted populations of peripheral T cells and incubate them with naive B cells for a couple of weeks, how well do they support B cell differentiation as measured by supernatant IgM, IgG classes. And what you see from these color-coded bars is that if you don't express CXCR5, you really don't support B cell differentiation. There's one crazy dove point up here that makes this bar go way up, but everything else is right here on the axis. So that you need CXCR5 to support B cell differentiation. But then within the populations, you get better and better B cell differentiation if you express CCR6 and best if you express CCR6 and PD1 in red. So in all cases, the red bars are the highest. So that's the population that appears to have the best differentiation of B cells. But once again, these are not pure populations. So if we look at these different populations and after 48 hours of stimulation, ask what cytokines are being produced, well, there's some interferon gamma being produced, mostly from this population. There's certainly a lot of IL-4 being produced there. Th17 cells producing IL-17 are very prevalent in these populations that we're calling potentially peripheral T follicular helper cells. So they're contaminated with T17 or Th17 cells. And IL-21 is actually produced by a lot of these different populations, but mostly by the CCR6 PD1 positive population. As Hendrik showed you and I showed you with HIV or SIV infection in the lymph node, you'll see accumulation of these cells. What happens in the periphery? Well, they don't accumulate. As you can see here in HIV-infected individuals with high CD4s or low CD4s, whether you just look at the CXCR5 high, the CXCR5 CCR6 high, or the CXCR5 high, CCR6 high, PD1 high population, they all go down. So they're not accumulating, they're actually decreasing. Now what people have found, however, and several groups are finding this, is if you just concentrate on this population, the CXCR5 positive population, and then use the other markers, what you actually see down here is that there might be a little accumulation if you say this is a T follicular helper cell population within the CXCR5 positive population. Then you can see a little bit of accumulation. And then with antiretroviral therapy, for all the different populations, you see them coming back, except here when you just look at the CXCR5 positive population, you see that you're not seeing those come back. You're not seeing them go down with antiretroviral therapy. So there's still some controversy over how well these mirror, looking at this population down here, how well these mirror what's going on in the lymph node. Another way of looking at these cells, however, would be to look at both the phenotype and their ability to produce IL-21. And I think this gets to be very important. So if we stimulate cells from individuals, and here I show a representative HIV uninfected, an HIV positive person on ART, or an HIV positive person not on ART, stimulate their cells with SEB, and ask, are there any CD4 T cells that look like peripheral T follicular helper cells by phenotype that also produce IL-21? What you see is in HIV uninfected individuals, IL-21 are in blue, and what you see is there are some that are CCR7 positive, CXCR5 positive, and CCR6 positive right here. But as you look at HIV-infected individuals, that population starts to decrease, and certainly among individuals who are not on antiretroviral therapy, there are very few cells producing IL-21, which are CCR7 positive, CCR6 positive, CXCR5 positive. And you can further look, this is with a nonspecific stimulation. How about HIV-specific stimulation here with GAG? Really, none of those GAG-specific IL-21 producing cells look like peripheral T follicular helper cells. They don't express CCR7, they don't express CXCR5, and they don't express CCR6. So there really does appear to be a loss of these cells with HIV infection. Now that doesn't mean that you can't stimulate these types of cells, these peripheral T follicular helper cells, with a vaccine. So Barney told you about our DNA Ad5 vaccine. That vaccine was never really set up to stimulate T follicular helper cells, but we decided to look within a trial at whether it did induce T follicular helper cells. At day 196 after DNA and Ad5, so that's 4 weeks after the last boost, we stimulated peripheral T cells with envelope. We gated on the responding cells, the central memory responding cells, and then looked at, among those, which ones made interferon gamma and which made IL-21. And not surprisingly, most of them made interferon gamma, but a few did actually make IL-21. And then we asked, well, what's the phenotype of those IL-21 producing cells? Do any of them look like peripheral T follicular helper cells? And actually, some of them were CCR7 positive, and some of them were CXCR5, CCR6 positive. So we were able to stimulate them. I would not want you to believe that the vaccine that we're testing was ever set up to do this, nor is this obviously the best response that we would ever want to have. But at least it shows you that you may be able to measure these in a vaccine trial. So I'd like to just finish up by saying that peripheral CD4 T cells, which can secrete IL-21 and support B cell differentiation, which have been termed peripheral T follicular helper cells, are contained within a population defined by CCR7, CXCR5, CCR6, and PD1. This is not a pure population, and there's overlap with Th17 cells, et cetera. These cells are really depleted during HIV infection, whether you define them phenotypically or functionally by the production of IL-21. And vaccination can induce antigen-specific peripheral T follicular helper cells, but I think that care really needs to be taken in defining vaccine-induced peripheral T follicular helper cells to avoid confusion in the literature, because I think too often people will say that they've got induction of peripheral T follicular helper cells by using a rather minimum definition of what that is. And whether vaccines that induce these types of cells offer an advantage over those that don't, I think still needs to be determined. And I would just like to say that although the work on the T follicular helper cells was really carried out by Kostas, Takuya, and Kristen in my group, with a lot of help on some of the work by Adrian's group with Adam Wheatley, and we obviously had help from a number of other individuals within the VRC and elsewhere. Thank you very much, and I'm happy to take any questions. Just shout, Rafiq. Oh, they're in.
So if you try now to combine the human work and the monkey work, so how would you interpret the loss of those cells in periphery? Is it they've been sequestered in the lymph node and they're not moving to periphery, or?
I mean, that's a very good point, and I think that that's an obvious interpretation, is that when you get accumulation of these cells in the germinal centers, that they're not getting out and getting into the periphery. There is some data with SP1 receptor that would suggest that's exactly what is happening. We also feel that probably what's getting out into the periphery is something before the germinal center T follicular helper cell. So as the cell is making the decision to go into the germinal center, it can either go to the germinal center or out into the periphery. And there may be a skewing of how that's driven during HIV or SIV infection. One thing we do need to do that has been brought up is we need to characterize these peripheral cells in monkeys where we can really look at those dynamics at the same time.
Thank you for the talk. In one of your very first slides, you showed CD95 as a marker for TFH. And do you know what's the function of CD95?
Well, for CD95, what we're using that for is simply to identify the central memory like cells, and then showing that within that population is where you find the T follicular helper cells.
It's me here.
Oh, yes.
In your SIV work, it seems that the help from the TF8 cells to B cells works pretty fine.
Mm-hmm.
But this week I saw a paper in the Nature Medicine that shows that the help for human cells, human B cells, are impaired because of the PD-L1 expression on germinal center B cells and the PD-1 expression on TF8 cells. So what do you think this is, that difference occurs?
Yeah, so I actually got in trouble at a meeting of T cell and B cell people when I mentioned that these co-cultivation assays of taking a T cell and a B cell and then just measure, 2 weeks later measuring supernatant immunoglobulin. I thought those were fairly crude assays, and the B cell people got rather upset with me. But I do think that, yeah, you can take these T follicular helper cells and you can mix them with B cells and you can show that they have a function supporting B cell differentiation. But there's obviously a lot more that needs to be done in order to get the type of broad neutralization that we want. And there may be impairments in some of those other signaling pathways. So yeah, from a very crude assay standpoint, they look okay, but if you start going deeper and deeper into it, I mean, by gene array, we know that there's all sorts of genes that are upregulated and downregulated, and so they're not functioning normally. And we're going to be learning over the years, what those specific defects are and which ones we may need to intervene with.
Okay.
Rick, can you say something? What we want is antibody in the serum or in the mucosal secretions. And what we can measure are B cells and CD4 T cells in the blood. But all the action is happening in the lymph node or in the bone marrow. So can you connect some of those dots and tell us how measuring things, cells in the blood will help inform us how well they're being somatically mutated in the lymph node, or how well they're getting into the bone marrow to actually make our antibody?
Boy, that's a big question. So yes, I mean, it would be nice to be able to look in the periphery just at the B cells and say, all right, with this vaccine or with this infection, we're getting the right type of B cells, and that's associated with long-term memory. Because what you're saying is we want to induce the right type of antibodies in serum, and we want those to persist. And that's going to require the right type of B cell, a long-lived B cell that gets a memory B cell or a plasma cell that continues to produce antibodies. Adrian McDermott is doing a lot, in my group, is doing a lot of work in this area, and I think he'll be talking about some of this in his talk on Monday. But it is clear that certain vaccinations will give you an antibody response that looks good, but then it just falls off. And we know from vaccination quite often you see these great antibody responses and then they go down. And that may be because we're not stimulating the right pathways to get long-term memory cells or long-term plasma cell responses, and there's a lot more that we need to do to figure out how to do that correctly.
In terms of this Dennis Burton monkey, that 88, has got neutralizing antibodies. Was there any difference in the disease or cure or?
So what I can tell you in the work that we've done subsequently with 9 monkeys where we do get some broad neutralization, the monkeys that develop the broad neutralizing antibodies tend to be the monkeys that have higher viral loads and more general immune activation. And I think what it's telling us is that if you want to get the kind of differentiation of antibodies that's required, you have to continue to stimulate the immune response. That immune response has to attack the virus, has to cause mutation within the virus, such that a new immune response can be generated that you escape from, and you need that to go on a lot. And so it's sort of a catch-22. It's not what we wanted to see, but when you really think about it, it's probably what's required. You need constant changing of your antigen and constant stimulation to get the degree of somatic hypermutation that we need.
But it's not beneficial for the host.
No, but I mean, if we can find a way of stimulating that type of response through vaccination, then they'll have those antibodies before they get infected, and they'll never go through that, and that's the goal.
So, Rick, one last question. So maybe I missed it, but in your adjuvant studies, I did not hear you say which adjuvant you would use.
Oh.
You were doing a trial, and whether there was any correlation between...
In our human trial or the monkey trial?
Well, in a human trial, let's say, based on what you've
done. Yeah, so I presented a monkey trial where we used multiple different adjuvants, and that was alum, TLR7, MF59, et cetera. The only human trial data I presented was DNA Ad5 without any adjuvant.
No, but from the monkey studies, if you had to translate that to human, would you choose a particular set of adjuvants?
Oh, I mean, I would choose one of them, the alum TLR7 or MF59, which seemed to stimulate better T follicular helper cells. Having said that, just inducing T follicular helper cells is not the end all and be all. We got to get the right antibodies out of that.
So have you gone back to the RV144 study and tried to measure the TFH cells?
For peripheral T follicular helper cells in RV144, no, we have not. But we hope to.